Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-Level Biology Practicals. This is Core Practical 7. Use a light microscope to make observations of sections of roots, stems and leaves. So in observing plant tissues, you have to make sure you wear gloves as well as eye protection. The tissues that we're going to observe here are going to be plant stems, plant leaves as well as plant roots. And because this part is about observing, we have to be able to draw the structures that we see. We also have to ensure that the cut pieces are as thin as possible. Of course, we're going to be cutting through stems, through roots as well as leaves. So the cut tissues have to be as thin as possible in order to visualize them clearly using a microscope. When we draw any structures, we have to draw only what we see, and we have to use an HB pencil, use a ruler with label lines or for label lines, and the label lines should not cross each other. The staining agents that can be used sometimes are toluidine blue. These can stain the lignins green. The pectins can be stained pinkish or purple or pinkish purple, and the nucleic acids can be stained green, blue or purple. Now this is still connected to microscopy, so we have to go through the procedure. You collect a piece of plant stem. It could be plant tissue, whether stem, root or leaves, and then you're going to place it on a white tile. The key thing is to make it as thin as possible. So you'll cut several transverse or across sections. Then you place the thinnest piece on a slide, add a drop of water and then toluidine blue. Allow 2 to 3 minutes. Gently remove the excess stain. Place the cover slip and observe under low power. Then draw and annotate an outline showing tissues of stems but not the details of the cells. You repeat this for another plant tissue, maybe it's a root or leaf, in order to make the observations. So in the exam, they could bring something like this. We can see tissues that are going to be observed like sclerenchyma, collenchyma, epidermis, xylem, phloem, cambium, and so on. So this is very common. The key thing is you have to observe in a microscope, draw, and do not include internal structures of cells because we are observing tissues and not just a single cell. So this brings us to the end of this core practical. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.